Speaker for the day, Dr. Loren Wold from The Ohio State University. And the V is very important. I understand how that works. He is a professor of nursing and medicine and also in the Department of Physiology and Cell Biology. He is the Assistant Dean for Biological Health Research. He has multiple R01s funded to support his research in cardiovascular areas. He does in vivo and in vitro work. He also is a fabulous mentor and has tons of student trainees and postdoc trainees and medical students who've come in and out of his lab. And we are very excited to have him speak today on, and I'm sorry, but I lost the title of your talk in an email chain. But thank you, Loren, and I will turn the floor over to you now.
Excellent. Well, thank you so much. First of all, thank you, Dr. Sewell-Lofton, Dr. Zhang, all the faculty for the invitation. In our Zoom world, it's amazing that we can do this without having to travel, and it's one of those we're making the most of it. So just want everyone on the call to know that we'll all get through this some way, somehow. So I'm going to go ahead and share my screen and just want to do a quick mic check, make sure you can hear me okay, and you can see the screen.
Yes, we can hear you and see your screen.
Awesome. So I did change the title, my apologies. I was going to talk just about our e-cigarette work, but that project is actually pretty new, and so I didn't have enough data to fill the whole time. So I wanted to give you a little bit more background of how we got there. And so I've changed the title a little bit, and the title now is Environmental Toxicants in the Heart. How bad are they? So thank you again for the invitation. And I think it's important to understand, we talk a lot about diseases. We talk, about diabetes and hypertension and heart failure. But I think it's important to remember how important just where you live actually is for your health. And so this really became apparent back during the London fog in the 50s, in the picture you can see here, where the pollutant levels were just astronomical. And these levels resulted in 12,000 premature deaths, as well as many patients going to the emergency room for sudden cardiac issues. And what I'm going to talk to you about is a field that I have lived in really since the inception of the field of environmental cardiology in the early 2000s. But I also want to talk about some of the self-induced environmental pollutants, some of our new work on e-cigarette use. And I'm sure your campus is a lot like our campus, where you walk around and you see students who are in faculty, patients who are using these devices. And I think what hopefully you'll appreciate from today's talk is they may not be as benign as the companies like to tell us. So the first part of the talk, apologies to Adam and Jay, you may have seen some of this, is talking about air pollution, which is an area I've lived in for a while. And what you'll see on the left is a picture in Beijing, China. And I think, China really gets a bad rap because people always say, well, China's polluted. Well, look at the right. That's actually Los Angeles. And I think you'll see the similarities between the two. And air pollution levels, which are now heavily regulated, for a long time really went unchecked. So what is air pollution made of? So air pollution consists of different gases, which I've listed here, some of the more important being carbon monoxide, sulfur dioxide, and ozone, as well as anything really of size fraction within the air. So liquid suspensions, and what we're going to talk about today, particulate matter. So what is particulate matter? Particulate matter has a carbonaceous core. It is surrounded by heavy metals such as iron, etc., organic molecules, nitrates, and sulfates. So it's a real heterogeneous mixture. And what you'll see here on the right is a coal generating power plant, which in Ohio, we have many of these, as many other states do. So these different types of fossil fuel combustors, if you will, produce particulate And particulate matter, we like to define into 3 categories. So PM10, which are considered coarse particles, these are around the size of a piece of pollen. They're 10 microns or so. PM2.5, which is what I'm going to talk about predominantly today, is the fine particulate. And that is produced by the combustion of fossil fuels. So planes, trains, and automobiles. And it's around the size of a red blood cell. And then the smallest of these fractions are the PM.1 or the ultrafine fraction. So what's interesting about the ultrafine or the size of a virus, I know we hear virus on TV now every night, and we don't like that word, but it's really an ultrafine particulate. And what's interesting about the ultrafines is they're actually sticky. And I'm hearing from Adam, it says swap views. I don't know how to swap view, Adam. Do we know how to do that? He says we're seeing presenter view. I'm not sure what that means.
Try your display settings up at the top left.
Okay.
Yeah.
Swap presenter view and slideshow.
There you go.
Oh, I just learned something. See, you can learn something new every day. I had no clue how to do that. Thank you. So getting back to it, so the ultrafine particulates are sticky, and so they can stick together and actually form fine particulates. So this really became important back in the early 2000s, and in 2010, the first publication, and actually last week or earlier this week, the AHA produced the second iteration of the scientific statement on environmental pollutants in the heart, which showed that the association between different types of mortality and percent increase related to all-cause cardiopulmonary disease, cardiovascular disease, and ischemic heart disease. So anything really here above this hatched line is a positive association, okay? And these are all independent studies that have shown that there's a significant association between mortality risk and cardiopulmonary disease, cardiovascular disease, and ischemic heart disease. So PM exposure has been associated with myocardial infarction, atherosclerosis, and vascular dysfunction, generation of arrhythmias, as well as heart rate variability. And this is important because still on days of heavy pollution, the number of patients that show up at the emergency department for sudden cardiac events continues to grow. Thrombosis and elevations in blood pressure, along with hypertrophy. So what does this all mean, and how do we study this? And so my lab is really interested in both the direct effects and the indirect effects. So the direct effects are the effects of those particles getting into the bloodstream, circulating throughout the body, and having an effect directly on the heart. Whereas the indirect effects are the effects through the lungs, for example. So the lungs, of course, are impacted by anything we breathe. And they release different inflammatory markers or oxidative stress particulates that can then themselves, those mediators, affect the heart. Both of these can cause cardiovascular dysfunction. So this is really where my lab falls, is studying the direct effects versus the indirect effects. And I'm not going to show you today any of the studies we've done on the indirect effects where we look at the lungs and what the lungs release into the bloodstream and how that affects the heart. We're really going to focus today on the specific direct effects. And so there's many different models that are used for instillation. There's tracheal instillation, nasal instillation, etc. What we use is actually inhalation. So we use a non-stressed environment where we expose animals and they basically breathe, not knowing that they're breathing heavily polluted air. So we're not inducing the stress that might be seen if you're doing tracheal instillation or other means. So in the first part of the study, we expose animals for a long term. We do 6 hours, or did 6 hours per day for 9 months. And on average, that averaged out to around 15 micrograms per cubic meter. And interestingly, that's less than what is considered safe by the National Institute of Standards. And this is our old contraption. I'll show you in a bit our new contraption, but this is where we're exposing our animals, and it looks kind of sci-fi, but basically, and I want to make sure you can see my mouse. Do you see my mouse? You may not see my mouse. Okay. But what you'll see there right around in the middle are two contraptions, and it's actually blown up in the lower right. This is where the mice are exposed. So the mice are in these little tiny boxes. They're running around. They're able to, they're not restrained. They're not held down. They're able to run around in a little cage. And they're actually breathing either filtered air or particulate infused air. And so the particles we actually take from the air in Columbus, Ohio, and we concentrate them. So what you'll see here, and this is for the first study that we'll talk about, this is the daily change, and it's graphed by week, of particulate matter levels in Columbus, Ohio. And what you'll see is we get to pretty high levels. So China, on a heavy polluted day will be 100 micrograms, say, for example. What you'll see here is with our concentrator, we're actually able to get to those levels in Columbus, Ohio. And this is really where it all started. So a funny story, because we've got a lot of students on the call, but I like to tell this story because I think says a lot about science, is we had been using a model where we were taking animals and we were taking the heart out, perfusing it with buffer that contained particles. And that's not really physiological, right? It was a modified environment that we created in order to expose animals. Well, serendipitously, I was at the Children's Hospital at the time down the road, and I got a call from a colleague there who said, we've got these animals that have been exposed for 9 months. We don't have any use for them anymore. What do you think about using them for some cardiac studies? They had done some behavioral studies. And I said, they're free. Why not? So we went and we got the mice. And, this, first of all, was a precious, these were precious samples. They had been exposed for 9 months. They had paid for these mice and for the per diems to keep them alive for 9 months. And we thought, let's do some studies on them. And lo and behold, we did echo on them and showed that just exposing animals to particulates for 9 months caused a significant decrease in fractional shortening, which is shown on the upper left panel here. And surprisingly, this had never been shown. So these were all just control animals that had been exposed, and they had a significant decrease in cardiac function. We did, and I show here a bunch of other measures, but this fractional shortening is really the one that we'll focus on. Well, my lab is a cardiomyocyte lab, so we actually take the cells out of the heart. So the heart is made up of cells called cardiomyocytes. The cardiomyocytes actually beat, so they contract and relax, and that's what causes the heartbeat. So what you'll see here, and I'm going to focus your attention on the bottom left PS panel, you'll see that in the FA versus PM 2.5, the solid bars, that there was a significant decrease in cardiomyocyte function in these myocytes isolated from long-term exposed animals to particulate matter. So not only was the in vivo cellular function impaired, but also the in vitro cellular function impaired. So we were seeing a persistent heart failure within these animals. So we also looked at calcium levels. So you may know from your physiology classes that calcium is a cycling ion that is involved and very important in cardiac contraction. And so what we showed here was that at baseline, there was no difference in calcium level. And that's shown in the lower left bars. And then on the right, you see what's called tau. Tau is the fluorescence decay rate. And so we load these cells with a fluorescent dye. That dye binds to calcium. And so when the fluorescence decays, that means the calcium levels are decreasing. And so there was no difference in calcium levels within these cells. And so we knew that there was a dysfunction and it was not likely caused by calcium. So, well, what does that mean then? Well, we were interested in all the different calcium proteins, which are involved in heart function. So what we see here is the raw blot, where we're looking at a multitude of different proteins involved. And the one that you'll see in the bottom right, phospholambin to circa 2A, what we showed was that in cells taken from the long-term exposed animals to particulate matter, we saw a significant increase in the phospholambin to circa ratio. And so that can really help us to design the studies that we're doing now, which is looking at what is the role of calcium or lack thereof in the dysfunction that we're seeing. Okay, that's great. But we also talked about inflammation before. And so we know that inflammation is involved in this process. And so another set of studies we did was we actually took serum that was collected 18 hours after the last day of exposure, and we analyzed for 38 different inflammatory markers. Here we showed, and here I'm just focusing on a couple of them, and I'll tell you why. So MCP1 and MCP3, which is myocyte chemoattractant protein 1 and 3, as well as the receptor CCR2. And what's interesting, we've done a whole host of in vitro studies. We had looked at just isolated cells in a dish and exposed them to different particles and stuff. And we've done all those studies. We always kept seeing MCP1 showing up. For some reason, it just kept showing up in all our studies. And here in our long-term, in cell homogeneous, we finally showed that MCP1 and 3 were significantly elevated. So in that first part of the talk, in the long-term exposed, we showed that normal levels of PM exposed long-term cause heart disease. And cardiomyocytes taken from those mice had reduced contractility that was altered through beta adrenergic stimulation, through potentially an increase in MCP1 and 3. So that's good and all, but interesting story, I was at the Children's Hospital at this time, and interestingly, I'd never done any studies in children. So I decided probably a good idea to do some in utero exposure. And so we knew that from the evidence epidemiologically that in utero exposure to PM caused an increase in infant mortality, low birth weight, as well as preterm birth. For example, one study published in Pediatrics, mothers in areas with high pollution had infants weighing 35 grams less on average. So was this due to the particulate matter directly? We don't know, but obviously there was a clear association. We also know that animal studies were able to recapitulate what was seen in humans. So we knew that diesel exhaust exposure prior to pregnancy decreased birth weight, increased abortion rates, etc., as well as function through an increase in myocardial lipid peroxidation. So this collectively is what's referred to as the fetal origins of adult disease, or the Barker hypothesis, which is exposure to toxicants at early age can potentially have an effect at adulthood. And so our question was, well, how does particulate matter exposure affect the cardiovascular system during development? So in these studies, we took and we actually bred animals. We bred, we put the male and female in the same, in the exposure chamber. And once we noticed a plug, we actually began exposure, and we exposed the moms every day until the pups were born. Once the pups were born, we allowed them to simply grow up to adulthood in room air. And we call this exposure the entire gestational exposure. So one set of studies I'm not going to talk about, which is the perinatal set of studies. And really where that was different is we exposed from birth, or I'm sorry, from conception, from the day we found the plug, all the way until weaning. So that was a different set where we exposed all the way through when the animals were nursing. And so we're not going there with this study. We're simply looking at the starting out with the in utero, okay? And what you'll see here, and this is just a foreshadow in the fractional shortening bar, the left-hand bar, in utero exposure, so stopped on birth, the data was the same as from perinatal. So it didn't seem to matter if we exposed during that time of nursing or not. We're going to get into that more in detail. So this is from some of our work published in JAHA. You'll see here the echocardiographic data, and in figure E on the lower left, you'll see the fractional shortening that I just showed you in the previous, coupled with perinatal exposure, but this is where these animals were just exposed in utero. We also see the PV loop data looking at contractility, and what you'll see here is we had a significant decrease in cardiac contractility in the pups, and mind you, these were assessed when they were adult, and so they were exposed in utero, and then they were allowed to grow until they were 3 months old. They were adults, okay? And also what I'll tell you here is we're just looking at the males, so the sons, if you will. And we saw in the, if you'll see in the PV loop, in the panel B, what we saw was a significant decrease in contractility. Well, what about the myocytes? So as I said before, I'm a myocyte person. So we did a bunch of studies and we looked here and saw the same sort of phenomena as we saw in the long term, which was a decrease in cell function from animals that were only exposed in utero, okay? And remember, assessed at adulthood. So we were also interested in looking at the electrophysiological parameters of the cells. And so we looked at what's called action potential duration. So basically how long that action potential lingers. So the action potential, as you know, is important for signal propagation, for signal transmission. And so we, as you'll see here, we looked at this at various stimulation frequencies, 3, 4, and 5 hertz, and saw that the action potential duration was greater in cells taken from animals that had been exposed in utero. So what that means is it took longer for the action potential to occur, okay? So that's interesting, but really why, right? Why is this occurring? And so interestingly, in the beginning, we were interested in collagen, which, as you may know, is involved in fibrosis or stiffening of the tissue. We show here various parameters looking at inflammatory markers as well as fibrosis within these cells, and show that lo and behold, there was a significant increase in inflammation as well as fibrosis. Slide, there we go. We also were interested in looking at the calcium contractile proteins. You'll see here SERCA2a. SERCA2 is a protein that's involved in contraction, sarcoendoplasmic reticulum, and it involves sequestration of calcium. And as I said, calcium is involved in contraction. And I'm not going to go through all these in detail, but what you'll see here is various changes in contractile proteins that could likely be one of the reasons that we're seeing that myocardial dysfunction. So another question we're interested in is, what about capacity to exercise? So part of these studies came out of a grant we have from the VA, where we're actually interested in developing a model of burn pit exposure. So you may have heard in the news about soldiers that are exposed to very high levels of pollution in burn pits. So those serving in Iraq or Afghanistan, they live close to areas where all their trash is burned. And so we thought, well, they're active people, they're exercising. Could exposure to burn pits actually have an effect on exercise? And lo and behold, it does. And what you see here is the total distance traveled, 1 week, 2 weeks, and 3 weeks. And in the light bars, these are the filtered air exposed in utero. And the dark bars are those exposed to particulate matter in utero. And what you can tell here is that the distance traveled is much less at each week for the animals that have been exposed in utero, and of course the average distance is much less. We also were interested in what if you did echoes on these animals after exercise, and what you see here is that at all of the weeks, wasn't significant at week 2, but it is less on average. The cardiac function was less in the animals that had been exposed in utero. And this stood through the study with exercise. Okay, that's great. But what we then wanted to do was start dissecting all of the different trimesters. So another set of studies that we looked at was exposing animals just during the first trimester. So just the first 7 days. Once the plug was found, we stopped after day 7, let the pups grow to adulthood, did the same measures as we saw in the previous studies. Lo and behold, we saw the same in vivo decrease in cardiac function, as shown here with fractional shortening, which was similar to the long-term as well as the entire gestation. We looked at, same slide, my apologies. So we were really interested, and I'm not showing you just because of time, all the other related parameters with cells, et cetera. What I'll tell you though is all of the different trimesters we looked at with exposure, we saw a similar phenotype to the long-term exposed. And so we decided to really get crazy and think about, well, what if mom or dad, mouse, what if mom or dad were exposed to pollutants prior to meeting each other? Could that have an effect on the offspring? And so, lo and behold, we did just that. We exposed mom and dad mouse for 3 months before mom and dad ever met. We introduced them with a nice dinner and a glass of wine. And guess what? We had pups. Those pups were never exposed to anything. They lived a charmed life in the animal quarters, breathing perfectly filtered air, ate and drank as much as they want. And we then looked at them at 3 months of age. And so what we're looking at really are there potentially any epigenetics going on with this phenotype? This was a paper we had in JAW in 18. And you'll see here, no change in birth weight, but body weight at adulthood, so at 3 months, as shown in panel B, was significantly decreased in the males that were exposed preconception, okay? So remember, just to reiterate, mom and dad were exposed together. I shouldn't say together. Mom and dad were both exposed before they met. We looked at the males, the sons, at adulthood. And what you'll see here are just various different weights and measures. You'll see here that heart weight to tibia length was significantly decreased in the animals that have been preconceptionally exposed to particles. Well, what about the hearts? And so look at panel C, you see a similar phenotype where we see that fractional shortening was significantly decreased. Very similar to if you expose animals to long-term particulates for 9 months. And these, mind you, were males who had never themselves been exposed. We looked at cardiomyocytes, and you can see here in panel A, we see a significant decrease in peak shortening or the contraction. So the cell contracts and relaxes. This was measurement of contraction, and we saw a significant decrease. A bunch of other parameters that I'm not going to talk about, but similar story to what we've seen before, which is a significant decrease in function. We looked at various calcium channel proteins. We saw very similar changes in circuit 2. So we know that the calcium handling is intimately involved in this process, as well as NCX, which was not significantly elevated, but was elevated on average. We also looked at the oxidative stress pathways as listed here, and we saw, and we're just showing you some examples, we saw a significant increase in NERF2, SOD, We saw a decrease in catalase, so oxidative stress pathways are also involved. Similarly, we were interested in inflammation because inflammation seems to be involved in everything in science. And so we decided to, once again, look at our multiplex assay. And what we're showing here are some examples. A whole host of inflammatory markers were significantly elevated. One thing I'll draw your attention to, which is that nasty MCP1, which we're showing down on the bottom right, which keeps popping up in every study we do. So we finally have started exposing MCP1 knockout mice because it keeps jumping in our face and hitting us in the forehead. And so I guess it's time we study it. So just showing you a representative plot here of MCP1. We also looked at, we've begun looking at the epigenetic markers, and this was in whole heart tissue. We saw a significant decrease in DNMT1. We also saw an increase in SIRT1 and SIRT2, which are very crude epigenetic parameters. I will tell you right now, we're also looking at the epigenetics involved in sperm gametogenesis, as well as in the oocyte, because it's obvious that the sons, as we assessed here, are getting this effect from mom or dad, and so we need to look at the gametes. So those are continuing studies. So just a summary of this first part, showing you, which I didn't show you all the data for perinatal, but remind you that that was exposure from when the plug was found until weaning, so all through nursing, the entire gestation or the intrauterine period, the first trimester, and preconception. If you look at the data between filtered air and particles, it's pretty similar. So it's obvious that particulate exposure is intimately involved in the progression of this type of heart failure. So we know PM is capable of causing contractile dysfunction by both lung-mediated and lung-independent mechanisms. Long-term is bad. Exposure during development can cause reduced cardiac function in adulthood, and it can also predispose individuals to developing cardiac dysfunction. So now on to the new stuff. So that's great and all, but you know that's living in an environment that can potentially affect you. What about a self-induced environment such as vaping? Okay, and a little bit of background. So obviously, vaping was developed, or at least the companies will tell you that it was developed for those who were trying to quit traditional smoking, right? As a smoking cessation tool. It contains various chemicals. The companies will tell you that it contains vastly less chemicals than traditional tobacco, but it contains propylene glycol, vegetable glycerin, nicotine, and flavorings. The combustion causes what we would consider hazardous inhalants. And so when you combust or burn this oil, it causes particles to be released into the air. Organic compounds, reactive aldehydes, nicotine, and heavy metals. So you're breathing those in. So companies like Juul, which you may have heard of, and these companies change daily. There's new companies coming out, those others that are leaving the market. They really focus their advertising on youth because it was deemed attractive to be able to do this, right? And I think that what companies and I want to think that companies were not targeting adolescents, but it was originally thought to be a benign, it's a stimulant, but who knows what the long-term effects are? There's likely none because there's so few chemicals. Well, alarming rates of use are occurring in youth. I tell a story. My sister is a 4th grade teacher in North Dakota. And weekly, she will actually confiscate these from students that are hiding them in their bags or have them, because you may have seen they look like little USB devices. and 4th grade. It's amazing. But look at this. This is a JAMA paper showing use of vaping products in 8th grade, 10th grade, and 12th grade. And these were students that were asked, have you within the last month used an e-cigarette? It's actually 27.5 is the latest study of 12th graders have used electronic cigarette products. So what do we know about the effects of vaping on the heart? Shockingly, not a lot. We know epidemiologically that there's reports of increased rates of heart attack among users. Several lung-focused studies have shown inflammation and a dampened immune response, and some specific cardiovascular effects such as cardiac myoblast viability decreases in vitro, increased platelet aggregation and thrombosis, as well as just damage to the endothelium or the blood vessel lining, which we all know can contribute to cardiac dysfunction. So, interestingly, as I said, very little is known about the direct effects of exposure on cardiac function in vivo. So what did we do? We took 1 month old, or what we're considering juvenile, around 13 years old in human years. We exposed them to either filtered air, PGVG, which is the solvent, and then PGVG plus nicotine, okay, 3 groups. This is our new exposure system, our little pie exposure. So you can see here, and I wish you could see my mouse, but you see the little cylindrical horizontal piece that looks like it's sort of opaque. And if you think about it, that's actually cigarette smoke or vapor within. And what happens is we make this oil, we combust it, and we pump 1 puff per minute for 4 hours a day, 5 days a week to these mice. So they're just hanging out in their little wedge in their cage, smoking all day, okay? So, and then we look at our various parameters that we've seen before in our PM studies, echo, etc. What you'll see here now is these are adolescent mice exposed for 3 months. And what you see in the left, this is the males, you see a significant decrease in fractional shortening in the EC, electronic cigarette plus nicotine, okay? So we saw significant systolic dysfunction in the hearts. Intriguingly, we didn't see the same significance in females on the right. We did see a decrease, but it was not significant. But shockingly, this is the first data, and we're just getting ready to publish this. This is the first data to show cardiac dysfunction from vaping in adolescents. We also looked at the E to A ratio. So on the top are the males, bottom are the females. And what we saw is a significant decrease in E to A ratio in males exposed during the juvenile or adolescent period. And this is reminiscent, this is what we look at for diastolic function. So not only did these hearts have systolic dysfunction, but they also had diastolic dysfunction. We've looked at a whole host of different proteins involved, and this is from whole hearts. We see here a significant increase in collagen 1, which is also coupled with some changes in the calcium regulatory proteins as listed here. So what was thought to be simply a benign novel product could have significant consequences. We also saw a whole myriad of changes in cytokines within the serum. I'm not going to go through these all in detail, but you can see some of the inflammatory markers, interleukin IL-18, CCL2, MIP1, et cetera, were significantly decreased, or increased in the nicotine group. And so vaping caused an inflammatory cascade, okay? We also were interested in, because as I showed you, females didn't have the same response as males. So we're interested in some of the nicotine metabolism changes that were hypothesized to be going on. And lo and behold, you'll see here that various breakdown products of nicotine are listed. And on the right is CYP2A6, which is an enzyme involved in the breakdown of nicotine, is much less in females than in males. And so potentially that discrepant sex-related difference in male versus female response could simply be due to a difference in enzyme levels. Some brand new data that I just got the other day from the group is looking at lung function in these adolescent mice. And this is looking at lung resistance, elastance, and tissue dampening. And what you'll see here is the nicotine is much less. So looking at the upper left, lung resistance. So the nicotine group or the green group is much less than the FA or the control group, the blue group, okay? So the nicotine group had much stiffer, if you will, alveolar space as opposed to the control. Interestingly, we also looked at this in adults. So we took adult mice, 3-month-old mice, in comparison to our 1-month-old juvenile, and we exposed them for 3 months. And what you see here is no significant change in fractional shortening. It was higher, but it was not significant. So we didn't see the same fractional changes in systolic function or diastolic function as seen by E/A ratio in the adults as we did in the juveniles. So pretty interesting thing to ponder, where if you think about the users, and one thing I forgot to note is most users of e-cigarette products are adolescents, or the largest number of users lately has been adolescents and not adults. So what's going on here? So what we see are that juvenile mice are more susceptible to electronic cigarette-induced cardiovascular effects. We're curious, are different stages of lung development more susceptible to exposure? So we could hypothesize that the adolescent mice, the lungs aren't quite fully developed yet. So we're causing this massive sort of inflammatory reaction going on within the lungs. And what are the effects caused by nicotine versus vapor alone? So as I said, our nicotine is produced and is combusted with PGVG or the oil. What about just nicotine alone exposure? So in conclusion, we saw a significant reduction in diastolic function in male mice, some sex-related differences in serum cytokines, as well as nicotine metabolites, and changes in cardiac proteins involving, including collagen 1, and some significant future questions that we're looking at right now. And one thing that I'll sort of leave you with, and then take some questions, is electronic cigarettes, and Juul in particular, and hopefully there's no Juul representatives on the call because they won't like me, but Juul changed how nicotine was delivered, and they began using what's called nicotine salt, which increased the pH of the liquid, which allowed smoking electronic products to be more palatable for adolescents. And you can see when Juul came on the market, the use went from 10 or so % of high school students to 27.5%. So, you know, these products aren't regulated. The FDA doesn't regulate them. They'll put warnings out, but they're not regulating their sale unless they ban them, which you've seen them do now with a lot of the flavorings. But these products are changing all the time, and I'm always shocked when I go to a gas station and there's a different new device out now. So thank you to the people that really did all the work here. I just get to talk about it. They always laugh at me when I come in the lab because I'll probably mess something up. Collaborators on the right, funding that we've been lucky enough to get. We also just a couple months ago got a really nice award from the American Heart to look at vaping, which is their ENACT program, or End Youth Vaping. American Heart has put tons of money into this. We got $5.5 million to do this, to look at how we can stop vaping in kids. And what I've learned with my lab is if you feed them, they'll do good work. And so this was at our last Christmas party we were able to have in person. So thank you to all those that did the work, and I'd love to take any questions.
Thank you so much, Loren. That was just an excellent talk and some really, really cool data. And my husband is a high school teacher, so I've been talking about Joule use with him for a long time. But I want to open the floor to questions from our students and other professors who have signed on. So just pop your question into the chat. Or if one of our professors wants to ask a question, you can unmute yourself and do that.
Adam's always got questions.
I always got a question. I think it's always the same one though, so I need a better memory. Excellent as always. I cut in and out of there because of my connection. After your DNMT and CERT regulation, have you got into any ChIP-seq or bisulfite sequencing on any of that?
Yeah, so Adam knows me well. I'm not smart enough for all those highfalutin studies. We just started. So Kristen Stanford, who we've been working with her, as well as a former colleague of hers who's at UMass, Oliver Rando, looking at the bisulfate and ChIP-seq in the sperm of the dads, and also looking at the hearts of the offspring. So we're really trying to understand. What we're trying to link is gamete epigenetic changes with cardiac endpoints. And it's proven to be difficult to really do that because it's very associative. But yes, so the short answer is we are actually doing those studies as we speak.
The thing I always like about your study design is you have the ability to answer a lot of time course questions that a lot of people just speculate at, because you have the gestational, you have the peripartum or whatever you
Perinatal,
call it. Yeah. So I look forward to that data.
Thanks, Adam.
We have a question from one of our students. She's asked, have you done research on the secondhand smoke exposure and the different gestational perinatal phases?
Yeah, so we haven't. The problem is these longer term studies are exceptionally expensive. And so, one could argue that at least with the e-cigarette studies, a lot of those would be considered a secondary exposure because they're whole body, and so they're just animals breathing in their normal environment. We're not actually, putting the cigarette, if you will, in the mouse's mouth. But no, so the short answer is we have not. And they're very important studies to do, and tertiary or third generation is also important, which is the re-aerosolization of particulates.
Hey, Loren, I have a quick question for you.
Hey, Martin.
Hey, how you doing?
Good, how are you?
Good, good. Really nice talk, as ever. I always enjoy listening to your talk. I have a question about transgenerational effects. So have you started to look at that? And if so, how many generations do you see this adverse effect of the PM 2.5 exposure on, I guess, it would originally be the grandparents or whatever. So how many generations does this go on for?
We have gone to F2s. So F0 or F0 are the initial exposed, where we're exposing the mom and dad. F1s are the children, and then F2s are the grandkids. So we've looked at the male F2s at baseline or non-stressed. They don't have a phenotype, but if you tack them or if you give them an MI, they are severely affected. So it looks like the F2s you have to stress. We're right now we had to restart our F1 F2s because we were just getting ready to breed the F1s before the pandemic, and then so we're a little behind on that, but we're actually not funded for that, and so we had to put that a little bit on the back burner. We've reinvigorated those studies now. It's just taking some time, but one thing that I'll say, and I've sort of stayed away from this a little bit today, is we don't see the robust phenotype in females that we see in males in most of our models. So I think a lot of that could be the age that we're looking at it, that they're premenopausal.
Yeah. All right, really nice. Well, thanks, Loren. I
Thanks,
appreciate it. And a follow-up question for that then is, for the males, I guess, when you said, if you tack them on F2, they're doing worse. So, this is a question I'm always wondering about. So is there any way to reverse these effects in subsequent generations? So if my grandfather was, working in a factory and he was exposed to PM 2.5, is there anything I can do to kind of improve my chances of reducing my risk of cardiovascular disease?
Yeah, it's a great question, Martin, and it's something we think about all the time. Is we can model things in mice pretty well, but humans are very bad at models of mouse disease. So how do we replicate truly what's happening? And I think our humans are spectacularly adaptive, and so I think that our bodies have adapted to changes from or fixed, if you will, changes from past generations. But I always tell people that a lot of this is as we see in many disease states, oxidative stress or inflammation driven, and so really any means to reduce inflammation or oxidative stress can only be beneficial, or at least not cause any ill effects. And so that's my long answer of saying I don't know, Martin.
Okay, thanks. I love it. Great. Thanks, Loren.
Thanks, Martin. Hope you're well.
And I just wanted to ask a kind of a clarification question about the mice that you got from the collaborator that had been exposed for 9 months. Was that 9 months of exposure from birth to when you started analyzing them, or so were they old mice? I guess.
Yeah, great question. So they had started exposing them at 3 months. So when we got them, they were 12 months old. So they weren't old. And that's something we struggle with in mice is, we think of PM as accelerated aging or environmental toxin exposure as accelerated aging, but it's difficult to dissect out the direct aging effects as opposed to particular effects. So good question.
Thank you. Any other questions from any of our students or faculty who are logged on right now? Trying to watch the chat. Well, thank you again, Loren. That was just a wonderful, wonderful talk, and we enjoyed having you today. And I know our students enjoyed getting to interact with you more informally before the presentation. So everyone, the worst part about virtual seminars is we don't actually get to applaud for you. You can do the little
exactly the thumbs up,
clap emoji, and thank you again for coming in. And
thank you everyone for coming on a Friday. I'm sure we got lots of Alabama football fans on here. I wish your team a good game. My team ain't playing this weekend, so awesome. Thank you so much. I appreciate it.
Thank you. If you want to stay on for just a couple of minutes, I can make sure Carrie doesn't have any administrative things that I'm not supposed to,
Yeah. Thanks, everyone. Have a great weekend.
Hi.
Hey,
All right. So I am going to send you an email after this. I should have already sent it, but I've been kind of a little cuckoo this week. Um, you will be receiving an honorarium for this talk.
oh, well,
So
that's lovely.
yeah, everyone loves money, right?
I love money.
Yeah, money. So I'm going to send you all of that information in an email that you will need to
Cool.
set yourself up so you can receive that check. My dog is doing something. Wilfred, what are you doing? How did you tangle yourself? Come here, buddy.
Well, she fixes that. I will say,
Aw, hi,
Did
buddy.
he
Oh.
tangled his leg around the laptop cord. I'm sorry.
Oh,
This
hi,
is
buddy.
Wilfred.
Hi, Wilfred.
aw.
Come on, buddy. Sit down. Sit down. Sit down. So I will send you that. Sorry, I'm sorry. I will send you that in an email. It's a relatively painless process.
Cool.
It's all automated now, so you just basically make a profile, and it's called iSupplier, and you just upload your W-9 and any relevant information, and then if you can just send me when that gets approved,
Sure.
you just let me know, and then we'll go ahead and get that taken care of for you.
Perfect.
Did Carrie talk to you about the recording before I logged on earlier?
That was another thing. Okay.
There you go.
So there you go. That was supposed to come with the iSupplier link. So we have a little form that you can sign if you consent or do not consent to recording.
No, recording's fine.